One of the major problems facing modern society is the vast amounts of waste it produces and how to manage them. They vary widely, but very often contain a large proportion of organic matter, mainly of animal and plant origin. Many industries, notably the agri-food industries, produce large amounts of organic wastes. Their disposal, and especially the treatments which reduce their polluting load, are generally very costly operations. Cities are faced with the complex problem of managing very large amounts of wastes, all of them containing a more or less high percentage of organic compounds. Agriculture also produces large amounts of wastes. Furthermore, on intensive stock-rearing farms, vast amounts of animal manure are produced. If discharged to the environment, it causes unacceptable degradation. In nature, the biological decomposition of organic matter in the presence of air, or more technically, in aerobic conditions, leads essentially to the formation of carbon dioxide and water. In certain natural habitats, though, oxygen may be limited or even lacking. In these conditions, which are defined as anaerobic, the decomposition of organic matter results in a gas constituted of more or less equal parts of carbon dioxide and methane. Because of its origin, this combustible mixture is called biogas. It is now clear that because of the vast and almost inexhaustible supply of organic waste, its transformation into methane can produce effectively an energy resource that is renewable, clean, and of high quality. This method of waste management also contributes to the conservation of the environment, provided it reduces and controls in an economical way the pollution generated. This film shows, in simple terms, the mechanism of the biological process which leads to the formation of methane. The technologies of biomethanation will also be demonstrated, with the significant results which have so far been obtained in the member states of the European Community. In order for the conversion of organic material into biogas to take place on any significant scale, other favorable environmental conditions besides anaerobiosis must prevail. Temperature values, for example, must fall between about 10 degrees and 60 degrees centigrade. Biogas is formed at a faster rate in the temperature interval between 30 degrees and 40 degrees centigrade, which is known as the mesophilic range. A second interval of temperature in which production is even faster is about 50 degrees and 60 degrees centigrade. This is the thermophilic range. The process also requires the presence of a minimum amount of water and an environmental pH around neutrality. The agents of methanogenesis are microorganisms organized in groups, each of which exerts a specific function. To begin with, the group of bacteria called hydrolytic disaggregates complex organic matter into smaller fractions. This action yields simpler molecules, which are soluble in the medium. At this stage, the same microorganisms, together with others, mainly fermentative bacteria, transform the solutized molecules into intermediary products such as alcohols, organic acids of low molecular weight, and volatile fatty acids, which include acetic acid, as well as hydrogen and carbon dioxide. Following this, another group of bacteria called acetogens begins to operate. They transform the alcohols and the acids into acetic acid, molecular hydrogen, and carbon dioxide, which add to that already formed. At this point, two distinct groups of microorganisms responsible for the formation of methane come into action. First, acetoclastic methanogens transform acetic acid into methane and carbon dioxide. Starting from the complex organic compounds, more than 70% of the methane is produced through this pathway. The other group, called hydrogenotrophic methanogens, utilizes carbon dioxide and molecular hydrogen to produce the remaining 30% methane. In so doing, they perform an action fundamental for the equilibrium of the process, namely, the removal of the hydrogen produced in the preceding stages. Any accumulation of hydrogen will block acetogenesis and cause an excessive accumulation of volatile fatty acids in the medium. If this happens, there is often an increase in acidity, which contributes to stop the process. Among the various microbial species present, we also find those which are called sulfate-reducing. As the starting material usually contains a minor amount of sulfates, these bacteria utilize a small part of the available molecular hydrogen to reduce it to hydrogen sulfide. Finally, we have a mixture mainly consisting of methane and carbon dioxide, with a small quantity of hydrogen sulfide. It is this mixture which is called biogas. What we have described are the essential stages in the mechanism of biomethanation, often called anaerobic digestion. We have shown them as a succession of different steps for the sake of simplicity, but in reality, all the stages take place more or less simultaneously. Biomethanation is a complex process. To understand it, research in microbiology and in biochemistry has been going on for years. But if we are to make practical use of it, we need a technology which will carry out the process in a most efficient and economical way. This field of research has therefore resulted in the design of various processes, which differ according to the type of waste to be treated and the nature of the organic compounds which it contains. The essential element of a process of biomethanation is the reactor, or digester. This is usually a cylindrical vessel in which the reaction we are talking about occurs. For research purposes and laboratory work, we normally use small digesters made of transparent material, so that we can more easily control and observe the process. The simplest digester, called batch, consists in this case of a simple bottle which is filled with dairy cattle manure. Mixing facilitates the conversion because it induces a better contact between the organic substances and the microorganisms. The process, being discontinuous, yields a production of biogas which is high at the beginning but decreases continuously to the end, with long mean residence times of the mass to be digested, which may take several weeks. Shorter mean residence times and more constant biogas production occur in digesters which are called completely mixed, in which the loading of the organic matter to be treated, the production of biogas, and the removal of the digested mass take place continuously. In this case, as the microorganisms required for the transformation are also removed with the effluent, it is evident that the rate of passage must be such as to permit the microorganisms to at least double their number in order to remain in sufficient quantity inside the digester. It is also obvious that the rate of transformation depends on the concentration of microorganisms in the reaction medium. To reduce the time of treatment, systems have been developed that are capable of maintaining the microbial biomass inside the digester for times longer than that spent by the waste flow to cross it. In contact reactors, for example, the microbial biomass is separated from the digested residue and recycled into the reactor. In this way, the increased concentration of microorganisms in the digester reduces the treatment time to only 4 or 5 days. Still shorter conversion times are possible when the microbial biomass is almost totally prevented from leaving the reaction medium. In this way, if appropriate inert carriers with a large surface area, made of plastic for example, are introduced in the digester, the microorganisms become stably fixed on these carriers to form a film. As the formation of gas occurs by passage of the effluent over the biological film and through the biomass in suspension trapped within the interstices, these digesters are also called anaerobic filters. As a further development, inert carriers of much more subdivided form, such as sand, have been utilized, which present a greater specific surface area. This allows for an equal volume of carrier to fix a higher number of microorganisms. The flux of waste and the continuous production of gas maintain these carriers in a fluidized state. From this, the term fluidized bed, by which these digesters are known, and in which the mean retention time of the organic substances is limited to a few hours. An equally efficient method of keeping the microorganisms in the digester is based on the capacity for the anaerobic community under certain conditions to form large-size agglomerates, real granules, which are heavy and consequently sedimentable. In these digesters with sludge beds, the waste to be digested is introduced at the bottom of the reactor and, moving upwards, passes through a thick layer of active microbial granules. The latter remain in the digester, whereas the waste flow crosses it with much shorter mean contact times. In recent years, this type of digester has made a decisive contribution to the use of anaerobic treatment for industrial wastewaters. Put briefly, we can say that the digesters in which the microbial biomass remains for a longer time than that required by the waste flow to cross it are particularly well adapted to the production of biogas from waste constituted by organic soluble substances. These reactors are also very promising for the treatment of very diluted organic effluents, such as urban wastewaters. But if one has to treat organic matter in suspension or in a semi-solid form, it is better to utilize batch or completely mixed digesters, which does not present the problem of excessive accumulation of solids in the reactor. For the treatment of these kind of wastes, however, it may be more convenient to operate in separate steps, in which hydrolysis occurs in a reactor distinct from the one in which methanogenesis occurs. Under this method, the optimum conditions can be obtained for each of the two steps of the conversion process. Research often requires work with very tall reactors. This produces the right hydrodynamic conditions for the conversion process. From the laboratory, where the various phases of the process can be carried out in a strictly controlled way, the next stage is generally the pilot project, which better represents real treatment conditions. A pilot plant is located near a poultry farm, for example, where organic wastes are available in an environment that fully simulates reality. The aim of these pilot plants is also to test and develop the most suitable hardware and instrumentation. On the real scale, the more traditional application of anaerobic digestion consists in the treatment of sludges from municipal wastewater treatment plants. In a plant like this one, only a fraction of the polluting organic matter is destroyed during the aerobic step of the treatment. Most of it concentrates in a sludge which can be separated from the water in these large settling tanks. From the treatment of the sewage wastewaters from the nearby city, an average of 2 liters of sludge per inhabitant is recovered every day. By anaerobic digestion, this can produce about 20 liters of biogas, 65% methane. The prevalence of suspended solids in the starting material suggests the use of digesters of the completely mixed type, operating at the mesophilic temperature of 35 degrees centigrade. The biogas is subsequently treated chemically to remove the hydrogen sulfide, which is toxic, and exerts a corrosive action on metallic parts. The treatment is also meant to reduce the carbon dioxide content in order to increase the calorific power of the resulting gas. The gas is now suitable for piping into the city gas distribution network. The anaerobic treatment of sewage sludges also reduces bad odors and the pathogenic germ content. Furthermore, by destroying the colloidal constituents, it makes dehydration of sludges easier, as well as its disposal in solid form. The anaerobic digestion of sewage sludges has also been utilized for small communities, where it is possible to use digesters prefabricated according to standard models. Here, the biogas is produced, and there it is burned in a boiler. The steam generated is injected directly into the sludge inside the digester to maintain the process temperature. A very important application of anaerobic digestion is the treatment of liquid animal wastes, or slurry. In intensive stock rearing farms like this one, which houses more than 2,000 pigs, these wastes are produced in large amounts and constitute, therefore, an important potential source of energy. Here, the system of washing and of effluent recovery becomes important. In this sense, it is best to use as little wash water as possible, because this will result for an equal amount of organic matter in a smaller volume to be treated, and also in a lower energy requirement to heat it to the process temperature. The digestion system utilized includes a reactor much like a contact, by which the manure of a 100-kilo pig produces about 120 liters of methane every day. Of particular interest in this installation is the battery of 10 co-generators which utilize the biogas to produce simultaneously electricity and thermal energy in the form of hot water at 80 to 90 degrees centigrade. The latter is obtained by heat exchangers which recover the thermal energy from exhaust gases, from engine cooling water, from the oil, and from the electricity generators. With these systems, the chemical energy of biogas is transformed into electricity and heat with an efficiency of more than 90%. Especially in stock rearing farms, the cost of anaerobic treatment of effluents is decisive in determining its profitability and consequently its success. This is why very simple and low-cost digesting systems were developed, like this one operating on a farm of 2,400 head of cattle. The digester of the type called plug flow consists of a single basin in which the manure, introduced at one end, pushes the mass already inside towards the outlet at the other end. The cover, constructed in supple polymeric material, serves also as gas holder for recovering the biogas. This digester, which is especially suited to the treatment of bovine manure, produces daily about 800 liters of methane per head of cattle. Also on this farm, the gas produced is converted into electricity and heat using a large cogenerator which functions with a high conversion efficiency of the biogas, close to 75%. A still simpler and cheaper solution is the one on this pig breeding farm. The plant consists solely of a floating balloon in supple polymeric material that collects the gas which is naturally produced in the manure storage basin. In systems of this type, the production of biogas depends strongly on the external ambient temperature, with a large output in warm weather and a reduced output in cold weather. But the system does not require any external energy supply, with the result that even in the most unfavorable conditions, whatever gas is produced, however little, is available for use on the farm. To achieve economies of scale, various biogas plants of a centralized type have been built in Europe. In this integrated complex, a network of underground pipes collects in a single treatment center about 900 cubic meters of manure daily, originating from 86 pig, cattle, and poultry farms spread over 36 square kilometers. Very interesting here is the use made of the 15,000 cubic meters of biogas produced daily in two large completely mixed digesters operating in the mesophilic range of temperature and in the post digester, which functions both as a gas holder and as a sludge thickener. Depending on the season and the needs of the moment, the gas is utilized for drying tobacco, for drying cereals and forage, and for heating 9,000 square meters of glass houses intended for growing flowers and vegetables. In addition, a variable supply of biogas feeds two groups of co-generators during the whole year. Once the energy demand of the center is satisfied, the produced electricity is sold to the grid. The liquid effluent of the digester is recovered in 2 large basins, from which it is redistributed to the holdings with a much reduced polluting load by means of a network of pipes for final recycling on the land as fertilizer. The digested solid fraction is dried and compacted to be utilized as soil conditioner. Besides the economic advantages derived from the energy utilization of the biogas, it should be stressed that this type of installation helps to conserve the environment thanks to a correct management of the large amounts of polluting manure produced on stock rearing farms. The anaerobic treatment of organic effluents of industrial origin has developed tremendously in recent years. Pulp and paper mills, distilleries, sugar refineries, breweries, meat processing industries, vegetable and fruit processing industries are all major potential utilizers of biomethanation systems. In this distillery, ethanol is produced by fermentation followed by distillation. The starting products are generally certain surplus